So, thank you for coming to our final learning workshop. I think it's like 12 or 13. This will be over proteins, amino acids, and enzymes. So we're going to jump right into this. I wanted to show you a little distinguishing between our amino acids. So there are four basic categories of amino acids that we usually kind of pay attention to. One is those with, and we group amino acids based on their side chains. And the side chains are highlighted in each of these four windows, okay? So you can see top left, the highlighted side chains are actually considered nonpolar. So the highlighted side chains, these are nonpolar in this top left region. The ones that have OHs on them or NHs or amide groups like a C double bond O NH, those are generally considered to be polar. So those are polar side chains. So either hydroxide or with an amide group. These are going to be polar amino acids. Then you have your basic amino acids, and it says they have a positive charge, although in truth, they only have a positive charge at certain pHs, if the pH is sufficiently high. But these amino acids in of themselves are basic amino acids. Lysine, arginine, and histidine are typically classified as basic amino acids. And then you have the acidic amino acids, which are the aspartic acid and glutamic acid. Like I said, they're not always negatively charged. The charge depends upon whether or what the pH is. So the charge depends upon what the pH is. And I'm going to go into that a little bit in just a moment. So I'm going to go ahead and stop sharing my beautiful picture. And instead, I want to show you something on the whiteboard. Okay, so generally speaking, as the name suggests, amino acids have two functional groups. They have the carboxylic acid functional group. They usually have a carbon here. And they have an amino functional group over here. Now, typically speaking, these amino acids always have one hydrogen here. But then the different amino acids have different R groups. And R just refers to like the rest of the molecule. You think of R as the rest of the molecule. Okay? An amino acid, amines tend to be basic. So this group here, tends to be basic. And this group over here is acidic. And so it turns out that in this form, you don't usually see amino acids in this form because the acidic group and the basic group tend to interact with one another. And usually what happens is the acidic group loses its hydrogen and the basic group gains the hydrogen. So amino acids are rarely ever found, or are rarely, if ever, found in this form that I've shown you here. Instead, amino acids tend to be found in this form here. The negative charge there. Your R group carbon. And the nitrogen with 3 hydrogens and a positive charge rather than 2 hydrogens. Now this particular form of the amino acid has a special name. It's called the Zwitter ion, the Zwitter ion form. And I have no idea why it's called the Zwitter ion form, what the word Zwitter means, but that's what it's called. This is Zwitter ion form. This is a much more common form of amino acids. Now, a couple things to note. If I slap this amino acid and if I put this amino acid in an acid medium, like a strong acid like hydrochloric acid, or even not as strong of an acid, but like vinegar maybe even, or lemon juice. If I put these amino acids in an acid, what happens here is this. What happens is that I will protonate, oops, sorry. You'll still have the 3 hydrogens on the nitrogen. That'll still be positively charged. The R group may be affected depending upon what type of an R group it is. If it's basic or acidic, etc. But in acid, the amino acid, in acid, the amino acid will be protonated on the carboxylic acid group, so this will no longer be a negative charge, it'll be a positively charged. Or rather, it'll overall be positive charge because that hydrogen is going to reattach to that oxygen because of the acid. Acid will cause that hydrogen to reattach. The acid will donate a hydrogen to that carboxylic acid group. On the other hand, if you were to have a base, if you were to react this amino acid with the base, the hydrogen, the extra hydrogen that's on the nitrogen gets ripped off, and this goes back to being NH2. So in other words, if you make the pH high enough in the solution, the hydrogen on the NH3 gets ripped off, and your overall molecule will be negative. If you drop the pH too low, make it too acidic, then the hydrogen will be added back onto the carboxylic acid group, and you'll get the structure on the left. If the pH is somewhere in that middle range, you're going to get this Zwitter ion form. Okay, so amino acids are the backbones of proteins, so the building blocks of proteins. How does that happen? Well, it happens through what's sometimes referred to as a condensation reaction. And I'm going to show you exactly what that condensation reaction will look like. So delete that. Let's say, for example, I have a C double bond O, single bond OH. Oh, I'm actually going to take that H off because that's not going to happen. It'll be in the Zwitter ion form in all likelihood. And then I have the R group here. And in this case, for the R group, I'm going to make it CH3. So this is just going to be a very simple amino acid, R group CH3. And then, and for those of you who are wondering, what does that make it? That makes it alanine. That's just alanine. So I got alanine here, and I have nitrogen, NH3 with a plus charge here. Well, if I combine that with nitrogen, there's 3 hydrogens here, positive charge, it's connected another carbon. I'm going to do a different amino acid. This time it's going to be a CH2 with an OH attached to it. And then I'm going to go ahead and do my carboxylic acid group. So this amino acid is called serine. So type this out. Alanine, serine, and no, you will not have to memorize these. All right. In a condensation reaction, if you think about condensation, what happens when condensation occurs? What forms? If there's condensation on a glass, what forms on the surface of the glass? Liquid what? Yeah, liquid water. So condensation reactions involves condensation reaction also involves the formation of water, 2 hydrogens and an oxygen. Basically what happens is I'm going to pull this oxygen and 2 hydrogens. So I pull an oxygen and 2 hydrogens. And I'm going to form water. And then the remaining pieces are going to stick together and be combined in one long chain. And so I end up getting this as my product. H3N plus a carbon, CH3, H, C double bond, O. But then instead of being connected to the oxygen, because the oxygen's gone, it connects itself to the nitrogen in the middle. And the nitrogen is only going to have 1 hydrogen left because it gave 2 away. And then I go to the other carbon, and it's going to have that H on top and CH2OH down here. And then I have the C double bond O, single bond O minus. So this is what might be referred to as a dipeptide. It's a very short peptide chain, a very short amino acid chain. It's composed of 2 amino acids, and typically when you see a peptide chain like this, sometimes they'll abbreviate it like this. Say, well, this is ALA dash SER. ALA for alanine, SER for serine. Sometimes they'll just abbreviate it like that. Now, the bond that I'm going to highlight in orange, the amide bond between the 2 amino acid chains, this is what's referred to as the peptide bond. So this is a peptide bond. The amide bond that forms between what was the carboxylic acid group and then the amine, that is the peptide bond. Now, amino acids can be linked together in very small chains, but they can also be linked together in very large chains. You can actually have amino acids that consist of literally amino acid chains, peptide chains that consist of hundreds and even thousands of amino acids. So when you have amino acid chains that are linked together and they're longer than a certain point, we tend to call those proteins. So proteins are complex amino acid chains, are large amino acid chains, and typically proteins have some sort of structure or function for which they are produced, so to speak. Any questions on this so far? Okay. So now I want to go over with you the 4 levels of protein organization, 4 levels of protein structure. I'm going to clear this out. So, primary structure of protein. So the primary structure of proteins is essentially just indicating what amino acids are connected to what amino acids are connected to which other amino acids. So if I have a polypeptide, like the ALA, SER, let's say you kept going, there's a glycine, GLY, there's a GLU, glutamate, glutamic acid. Let's say there's a cysteine, CYS, etc., etc., etc. Just that listing of the amino acids, you could also draw them out, but that would take a really long time. But the primary structure of proteins is basically this. All it is the order in which the amino acids are bonded. So the order in which the amino acids are bottom from start to finish. And actually, I'm going to go ahead and type that out. So the order in which amino acids are bonded from start to finish. When I say from start to finish, what I mean is from C terminal to N terminal. Okay, so what does that mean? Well, let me go ahead and, for the purposes of our discussion, I'll redraw what we had previously. You had the C double bond O minus, and you had the carbon, hydrogen, and we had the alanine, which is a CH3 here. And on the other side, you have the NH3. Now, let's say that, like I said, it doesn't stop here because they're connected to several others ones. So I'm going to go ahead and do a few of these connections. I'll do the first 3. Let's just say we had the amino acids chain stop at 3. Okay. So in that case, this would be an NH, and then C side chain serine is going to be CH2OH. Next, C double bond O. Of course, there's the hydrogen up here. Oh, I lied. No C double bond O because I'm going to do one more chain. Won't have an H anyway. I'm going to have to get glycine. So connected to another NH. C. Glycine side chain is just another hydrogen. Very simple. And then I have a C double bond O. Oh my. Okay. So in this particular structure, in this tripeptide, I have an amide bond right here, and I have an amide bond right here. Those are your 2 peptide bonds. and they separate the different amino acids. I have alanine, ALA, I have serine, SER, and I have glycine, GLY. And here's the thing. Notice that the two ends of the chain are not the same. On one end of the chain, I have a NH3. On the other end of the chain, I have an O-. You will always have this in any amino acid chain. You're always going to have a nitrogen at the end of one end and an oxygen at the end of the other. So the area where at the end of the chain that has the nitrogen is called the N-terminus. That's the N-terminus. That's the amino acid at the end that terminates or ends with the nitrogen. And then on the other end, you have the C-terminal amino acid. Sometimes it's called N-terminus. Sometimes it's called the N-terminal amino acid. Just so I'm using the same terminology as your book, I'll go ahead and write N-terminal amino acid. This is also called N-terminus. And this is going to be the C-terminal amino acid on the right-hand side. C-terminal amino acid. I have the N-terminal amino acid and the C-terminal amino acid. The N-terminal amino acid and the C-terminal amino acid in the chain. So the primary structure of the amino acid, primary structure of proteins just indicates what amino acids are bonded to each other. And on one end, you'll have the N-terminal amino acid, and the other, you'll have the C-terminal amino acid. So the primary structure of proteins, this is held together by peptide bonds. And that's important to note, so I'm actually going to type it up here. Held together by peptide bonds, because you're going to be expected to know what holds each of these levels of structure together and what can break them apart. Right? So this is primary structure of proteins is held together by peptide bonds. We'll deal with the denaturation of proteins later on. All right, let's go to secondary structure of proteins. Secondary structure of proteins. Actually, I'm going to clear this because I want more room. Secondary structure of proteins. So what is the secondary structure of proteins? Typically speaking, the secondary structure of proteins has two different forms, the alpha helix and the beta pleated sheet. And both of them kind of work the same way. Okay? So, what holds these groups together? Well, it turns out that what holds these groups together are actually the interactions between the hydrogen bonding interactions between the nitrogen, the NH bond, and the carbonyl. So I'll show you how that works. So let's say, for example, I have amino acid, okay? I'll draw my amino acid. So I have, let's see, a C double bond O. And it's, let's say it's connected to a nitrogen up here, okay? From a different amino acid. But C double bond O, here's your side chain, R, and then you have a nitrogen attached to a hydrogen. And then you have a C double bond O, the carbon that's attached to a side chain. And then you have the nitrogen attached to hydrogen. And then I have a C double bond O, a carbon attached to an R group, and then I have a nitrogen attached to a hydrogen. You should be kind of seeing at this point that this pattern is kind of like continuing. All right, and so it kind of keeps going down that way, and then it comes back up. And I have a carbon double bond O. And then I have a carbon with an R group and a hydrogen. And then I have a nitrogen with a hydrogen. And then I have a carbon double bond O. And then I have another carbon, the R group, and a hydrogen. And then I have a nitrogen attached to the hydrogen. And then I have a carbon double bonded to an oxygen. Carbon with the R group. And a nitrogen. So the secondary structure of proteins, it's really hard to separate because it involves a lot of different attractive forces. So, for example, here, if you see this, what happens is that there's an interaction between the lone pairs of electrons on the ketone group, or on the amide carbonyl, the oxygen here. There's a attraction between the lone pairs on those oxygens and the double bond O and the hydrogen that's attached to the nitrogen. In fact, these are hydrogen bonding interactions, because anytime you have an NH, an FH, or an OH, you have hydrogen bonding. Now the nitrogen provides the NH bond here, but the oxygen provides the electronegative atom from another molecule. So you have these hydrogen bonding interactions, and I'm actually going to show these hydrogen bonding interactions in a yellowish color, but they're pretty strong. And notice how many of them there are. Notice how frequent they are. Those hydrogen bonding interactions are all over the place, right? So these are very, very strongly held together into these secondary structures. And if they're twisted around each other, you get this what's known as an alpha helix. But if it just goes up and down like this again, then that gives you what's known as a beta pleated sheet. So up and down. You have these amino acid changes kind of like going up one way, down the other, up one way, down the other, up one way, down the other. That's a beta pleated sheet. And so you get the interaction of these. Notice that the key point of the key point here with the secondary structure is that it's the structure is structure created by the interaction between, and actually specifically by the hydrogen bonding interaction between the carbonyl and the amino/amide nitrogen for every on each amino acid. So the structure is created by hydrogen bonding interaction between the carbonyl and the amino acid and H on each amino acid. So that's where the alpha helix and the beta pleated sheet come from. Any questions on that? Okay. Cool. And I apologize that this isn't as much of an interactive session, but most of this chapter is primarily terminology. And I thought it would be helpful to kind of break it down for you a little bit better this way. So this is the secondary structure of proteins. Now the tertiary structure of proteins is interesting. The tertiary structure of proteins is interesting because it involves very similar things, except, well, actually, I think I'm going to clear this. Okay. So tertiary and this is the structure of proteins that forms due to the various interactions of the amino acid side chains. So effectively, you know how we had the you know how we were doing the whole thing where we had the the C double bond O, etc., and then the nitrogen NH, and then the C R group, and then you have the C double bond O NH, and then the CR group. So it's the R groups, the side chains, that are responsible for the tertiary structure. It's the R groups or the side chains that are responsible for the tertiary structures. So just like we have, if this was to kind of go over here and come back up here, you know, you might have the C double bond O, and you'd have the NH here. For the secondary structure, you got H here, etc. Oh, wait, whoops, sorry, not H, sorry, C, R group, H, C double bond O, etc. And when this comes back down the other way, these R groups can interact with each other. These R groups can interact with each other. And because the R groups can interact with each other, it ends up leading to the creation of what are known as tertiary structures. Now, the tertiary structures, the interaction between, and I'll actually go ahead and circle this. So this is, but this doesn't happen as frequently because, I mean, you're not getting as many interactions with these R groups as you are hydrogen bonding interactions between the amide NH and the carbonyl. But these do end up causing important structural changes to the protein, and in fact it causes the protein to wrap around itself, sorry, this is a carbon, wrap around itself in a particular way. And I apologize, my writing is a little bit sloppy, but so it's the R group interaction that's responsible for the tertiary structures. And there's basically 5 different types of interactions that are responsible for tertiary structures. So the 5 types of interactions, 5 types of R group interactions that are responsible for tertiary structure. The 5 different types of R group interactions are responsible for tertiary structures. Here, put a big line. There we go. So what are they? Number 1, hydrophobic interactions. Number one is hydrophobic interactions. So remember I showed you how the different R groups, some of them were non-polar. Like the ones that had the CH3, like alanine, that's a non-polar group. Anything that's just composed of carbons and hydrogens, those are going to be non-polar. Those are going to be non-polar side chains, and they're going to interact in such a way as to give you hydrophobic interactions. So it'll be attractive interactions between non-polar groups. So attractive interactions between non-polar side chains. Then we have hydrophilic interactions. And hydrophilic interactions are kind of interesting because this is kind of an interaction of the protein with the solvent around it, with the fluid around it. Keep in mind, obviously, that if you think about, like, for example, if you think about the proteins that exist in our body, well, what do we have in our body? Primarily, I mean, blood. I mean, technically speaking, we also have a less polar fluid called lymph, and of course we have fat and such. But hydrophilic interactions are in some sort of aqueous media, some sort of water-based media. Well, this is the attractive interactions. Attractive interactions between polar R groups, polar side chains, active interactions between polar side chains, and realign that, and the polar medium. And what this causes polar side chains to try to migrate to the outside of the protein structure. And that allows it to interact with the fluid on the outside. It allows it to interact with the aqueous medium. That's an example of the protein structure. So essentially, there are attractions between the external aqueous environment outside of the protein structure to interact with the external aqueous environment. Okay, that's basically hydrophilic interactions. So then there's the salt bridges. So you'll notice that when I showed you some amino acids have side chains, some amino acids that have side chains that have carboxylic acid groups, right? And some of them have amino groups. So if your R group has an NH3 somewhere or an NH2 plus, that's positive. And then if there's another amino acid side chain like glutamic acid that has a CO, C double bond O, single bond O minus, those positives and negative charges attract one another. And so that leads to what's referred to as a salt bridge. A salt bridge. So the salt bridge is basically ionic bonds. Ionic bonds are interactions between acidic and basic amino acid side chains. Acidic and basic amino acid side chains. That's pretty much it. So the positive charge on one attracts to a negative charge on the other. Okay, and then number 4 is hydrogen bonding. And notice this is distinct from number 2, because the hydrophilic interaction involves the interaction of the amino acid side chains with the external aqueous environment. In other words, with the cytoplasm or the fluid surrounding the protein. Hydrogen bonding, though, refers to the interaction between polar side groups. And so this is basically attractive interactions between polar side groups, such as those with OH groups or amide groups. So the side chains that have OH groups or amide groups, the ones that I showed you that were polar, they can be attracted to one another. They can be attracted to one another because of hydrogen bonding. And those hydrogen bonding interactions between the R groups would be like the ones we show here. The R groups are interacting with each other, whereas the hydrophilic interactions would be the R group interacting with the exterior solvent. All right, one more to go, and that is the last factor that is responsible for tertiary structures, and that's disulfide bonds. And this is really limited to one specific amino acid. So basically two cysteine groups will react with one another to form a covalent bond, a covalent sulfur-sulfur bond. So the disulfide bonds are actually covalent bonds. So it's not just an interaction or an attraction, And so these cysteine groups, even though obviously cysteine is only one of 20 amino acids, just a few of these amino acids, just a few of these amino acids that link up in a protein structure can add significant strength to the tertiary structure because of the fact that they're covalent bonds. So these are the 5 types of R group interactions that are responsible for tertiary structures. And these tertiary structures are ultimately what give the protein its function. The tertiary structures are responsible for allowing the protein to have the specific function and ability that it does. For this purpose, let's see if I can find a good picture of tertiary structures. I want to show you. All right, so this shows some examples of tertiary structures. So as you see, here's your hydrophobic interaction in the pink. 2 carbon-hydrogen groups interacting that attracts each other. You then have a hydrogen bonding interaction a little further to the left, where the OH from a serine interacts with the oxygen from carboxylic acid chain. That's a possible hydrogen bonding interaction. You can also have disulfide bridge, the one in yellow. The 2 cysteines, they lock those 2 sides of the chain together and force it into a loop. And then there's also the salt bridges, which shows the ionic bond between positively charged amino group and negatively charged carboxylic acid group. So yeah, this is an example of tertiary structures due to the polar R groups. Now, of course, it doesn't show the hydrophobic or hydrophilic interactions with the polar groups migrating to the exterior so they can interact with the aqueous environment. But that's also, you can assume that would also be present on the outside edges of that. Okay. And then last but not least, let's do quaternary structure. And I won't spend a whole lot of time with this one. Quaternary structure. Essentially, a quaternary structure is a larger structure formed from 2 or more proteins whose tertiary structures combine to form a larger, typically biologically active, complex protein. A larger structure formed from 2 or more proteins whose tertiary structures combine to form a larger biologically active complex protein. So quaternary structures basically is just 2 or more tertiary structures combining together to form a super large molecule. And quaternary structures are held together by the same things that hold together, these are held together by the same interactions that hold together tertiary structures. Hold together. So same interactions that hold together tertiary structures. all the side chain interactions. The only difference is that a quaternary structure is formed from two or more separate tertiary structures, two or more separate amino acid chains, two or more separate proteins that combine to form like a larger protein. Multiple polypeptide chains, basically. Okay, so I want to go through a couple other things with you real quick. Well, not necessarily super quick, but now we have to deal with protein denaturation. So what can destroy a protein structure to make it non-functional? Because the reason why these tertiary structures exist, the reason why you have the tertiary structures shaped the way they do, is so that they can form a particular function in the body. If they're not shaped the way that they are, if they don't have the tertiary structures that they do, they can't work in the body, they can't function in the body. The body is not able to make any sort of use of that protein, so to speak. So there are some things that can happen which will render a protein useless by destroying either different levels of structure. Okay, so so these are the different 5 different ways that you can denature a protein. So um, let's see, let's do make a line. So so this is so this is the denaturation agent, denaturation. Either's fine. But this is what causes it to denature, to break down. First is heat. Okay? Usually above 50 degrees Celsius, but sometimes you have to go significantly above that even. 50, huh, 50 degrees C. And so what does that do? Well, and so what does it do? Uh, well, when you heat something up, they move faster, okay? They move faster, they're more active, they're more energetic, they're harder to keep together. It so destabilizes hydrogen bonds. It stabilizes hydrogen bonding interactions. It destabilizes hydrophobic interactions. Hydrophobic interactions. And at high enough temperatures, and for a significant amount of time, uh, you can end up even disrupting the primary structure. You can end up causing hydrolysis or the splitting of the peptide chain. But typically, most of the time, you don't get to that point. Usually, it's hydrogen bonding with hydrophobic interactions, hydrogen bonding interactions and hydrophobic interactions that are disrupted. And so, what are some examples? Biggest example is cooking food. So cooking an egg, you can see the egg gets solid. So the egg gets solid because the proteins are denatured. Okay, so what's another one? Let's do. Does anybody know another denaturation agent? What's another thing that can cause your proteins to denature, other than heat? Force depends on what you mean. Shaking. Okay, that's true. Agitation. Agitation. Violently shaking or disrupting it like that. And what does that do? Well, it disrupts. It's fine. Good idea though, but it disrupts hydrogen bonding interactions. It also stretches. It disrupts. It also disrupts the hydrophobic interaction or hydrophilic interaction. Hydrophobic, sorry, hydrophobic interaction, and it stretches the. It can stretch the peptide chain, stretch out the structure. Okay, so an
Ryan,
example of this. Yeah, go
switch
ahead.
off the gas, the stove. Switch it off.
An example would just be like
Oh, sorry.
whipping cream. No, that's okay. Example of this would be whipping cream. Another example would be uh, whipping up egg whites to form meringue. I don't know if I know how to spell meringue. Is that right? I don't know if that's right. Meringue. I think that's right. Maybe it is. Okay, so whipping up egg whites to form meringue for like a lemon meringue pie. All right. What's another one? Does anybody have another idea of something that could denature a protein, make it useless? Acids and bases. That's right. So acids and bases will also do that. Let me change color. I'll do orange this time. Acids and bases. 3. Which is why sometimes you'll notice that for certain dishes, like for example, there's a type of Hawaiian fish, uh, a type of Hawaiian fish dish that it's not cooked with heat, it's cooked with acid. It's basically raw, but it's cooked in acid. Or they call it cooking it with acid. But it's called poke. You might see poke bowls and things like that at certain Asian restaurants or Hawaiian restaurants. But basically, acid and bases also uh, disrupt hydrogen bonds, and they also they disrupt the hydrogen bonds between polar R groups, disrupt hydrogen bonds between R groups or side chains, and not only do they do that, but they also disrupt salt bridges. If you think about it, if it's basic, it's going to protonate that amino. It's going to protonate that acid, and therefore the acid will be neutral. It won't be negatively charged. You won't have the C double bond O minus. You'll have a C double bond OOH. So it disrupts salt bridges as well. It disrupts hydrogen bonds and it also disrupts salt bridges. Messes up salt bridges. Okay, and there's another one. There's a couple of others. There's 2 more. What are some other ones? Other ways to denature proteins? Well, I'll tell you. There's one. One is heavy metal ions. Heavy metal ions disrupt salt bridges as well, and they also destroy uh, disulfide bonds. uh, destroy disulfide bonds, and they also disrupt salt bridges. They're not spectator ions, they actually lock themselves together with um, whatever carboxylic acid group they'll combine with. Heavy metal ions are positive, so they'll combine with those negative carboxyl groups and lock them down and prevent, you know. So that's where mercury, that's why mercury and lead are so dangerous, mercury and lead ions, because that gives you, they can poison you like that, mercury and lead poisoning. What it does is essentially it disrupts and screws up the proteins inside your body. And the heavy metal ions are kind of hard to get out too, because they start to get lodged in your system, and it can be really very much a challenge to remove. Okay, and then of course there's also lactic. So for acids and bases, you can have um, adding lemon juice to milk to make ricotta cheese. Ricotta cheese can be is a very simple cheese to make. It basically is just adding an acid to the milk and letting it curdle out. Make ricotta cheese, adding lemon juice to milk. It denatures the proteins and causes them to curdle out.
So does it mean that the protein doesn't have any nutrient? There's no nutrient there anymore when it denatures, right?
No, no, and that's not what it means at all. Because when you cook the proteins, it's not that your body can't use them in terms for food, because what proteins that your body eats that goes through digestion get broken down into their respective amino acids anyway, and then it can be processed that way for nutrition for your food. Keep in mind, those are foreign proteins anyway. Those are, like, for example, if you eat meat or you eat vegetables, those are plant proteins or animal proteins that are foreign to your body. So it's not like those proteins would be not like those proteins would be proteins that we would want in our body doing things for our body. They have to be, when you eat them, they have to be broken down and into amino acids and then used by our body later on to form our own proteins. But our body takes care of forming the proteins that we need. But we eat protein for the raw material, for the amino acids, and we can also actually process them for energy too. But proteins don't process it, into ATP, the energy molecule, very easily. And there's some not so great side products when proteins are forced to be used for energy, which is why the Atkins diet, though effective at helping people to lose weight, actually caused a number of other fairly serious health complications, because people were eating tons and tons of protein, but not a whole lot of anything else. In truth, for our body, fat is probably the cleanest burning fuel for our body. Fat is, believe it or not. Carbohydrates work. They can provide energy to the body. But fats are probably the cleanest burning fuel for our body in terms of not creating undesirable side products and side effects. So there you go. Now we have one more to go, and one more thing that can actually do it is nonpolar compounds or organic compounds. So let me change the colors once again. What color should I use? I'm going to use purple. Why not? So the last thing that can denature proteins is um, organic compounds. In other words, nonpolar usually, or compounds that at least have a significant nonpolar component to them. Because these compounds that have a significant nonpolar component to them, I mean, they can't even be polar compounds, but as long as they have a significant number of carbons and hydrogens, they can disrupt hydrophobic interactions. They disrupt hydrophobic interactions. So, disrupt hydrophobic interactions. So an example would be, what's a good example? I mean, obviously, if you know, benzene is toxic, you don't want to eat any of that. Most organic compounds are poisonous and or carcinogenic. 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 I don't know how to spell carcinogenic. Oh well. Cancer causing. Alcohol can be used to disinfect surfaces or skin for like injections. Why can alcohol be used to disinfect surfaces? Well, because it causes the denaturation of the proteins that the bacteria can use, and structure that kills bacteria because it messes with their it messes with the hydrophobic interactions of the proteins that they rely upon to function and survive. So yeah, so those are the 5 different ways to mess up a protein, basically. What bonds they mess up, and some examples. And to be honest, I don't really have much more to go over with you. The only other thing that I was going to mention is talking about enzymes and briefly referencing the different types of models of enzymes and types of inhibitors. So, is it okay if I erase this? Are y'all cool with this? Okay. Um, let's go ahead. Oh, I don't know why I stopped sharing. Here we go. So, enzymes are proteins that are shaped basically in a particular way. So they're shaped in a particular way so that they can match a particular compound. So I'm going to draw my squiggly protein here. So this protein is shaped in a particular way so that it can fit a very particular substrate. The substrate in this case, you know, might be, say, a molecule that looks like this. Okay. So I take my little protein. Um, yeah, I probably could have drawn this more simply. I think I will next time. Okay. And so yeah. So basically when the substrate is meant to fit exactly into the active site, so this here is your active site. Active site. And you have the enzyme, which is the bluish thing, and then the red thing, which is your substrate. That's basically the molecule that's going to be reacting. Essentially, enzymes are catalysts. They're biological catalysts that help a reaction to occur more quickly. So enzymes are biological catalysts that help a reaction occur more quickly by binding, holding a substrate. And making it easier for two or more molecules to come together to combine and react, or make it easier for a single molecule to break apart. Essentially, most of the reactions that are necessary to keep us alive would not be able to happen quickly enough to keep us alive if it wasn't for enzymes. So the enzymes, these proteins and such, that do all the hard work of helping these reactions to go faster so that we can actually have the energy needed to walk around, talk, and do all the things that we do. They're fueled by these biological catalysts, these enzymes. So just to give kind of like a rough and very simple description, if I had like, there's two different types of models for enzymes. So this is referred to if for the lock and key model. Lock and key model, enzyme is shaped exactly to fit the substrate. Enzyme is shaped exactly to fit the substrate. That's the lock and key model. So essentially you go from enzyme substrate, and it's kind of the model that I had above, if my drawing was worth a darn. And then this is supposed to be kind of a circular thing. I know that didn't actually work out like I had hoped it would. So it fits perfectly into the circle that is not totally a circle. Okay, so that's the lock and key model. The other type of model is what's known as the induced fit model. Let me go ahead and move this. Enzymes, biological catalyst, lock and key model, I'll put that up there. Now, I'm going to do the induced fit model. Induced fit model. In the induced fit model, basically, the enzyme adapts to fit certain, but not all, substrates. There are certain substrates that the enzyme is designed to fit. It will only arrange itself to fit those substrates. So like, for example, that would be like in case if it was like something like this. And let's say the substrate that you had was a little bit more narrow. Okay. So this might, when it senses the substrate coming near, it might narrow itself so that it fits the substrate, like clamp it in. It didn't fit initially, or it wasn't an exact fit initially, but it's designed such that it can kind of fit around it if the substrate gets near it. The enzyme adapts to fill certain but not all substrates. Okay, so those are the two basic models of enzyme interaction with substrates. Now we'll speak real briefly about inhibition, and then we'll be done. I'm going to go ahead and delete this. Are we good with this, or do you need any more time? Okay. Okay. So, inhibition of enzymes. So, enzymes, it turns out, only work optimally at certain temperatures. Interestingly enough, so enzymes tend to work. Enzymes tend to work best at a pH of roughly neutral, maybe slightly basic, about 7.4. Which, guess what, is roughly the pH of our blood. Enzymes also tend to work best around 98 degrees Fahrenheit, or 37 degrees Celsius. Which, guess what? That's the temperature of our body. So the reason why our bodies work at the temperatures they do, and the reason why they keep themselves at the pH that they do, is to maximize the efficiency of enzymes. Because the enzymes are what do all the hard work of carrying out chemical reactions to keep our bodies functioning, healing and repairing itself, and stuff. So enzymes can be inhibited by either competitive or non-competitive inhibition. So competitive inhibitors compete for the active site. So competitive inhibitors are non-active substrates that compete for the active site, blocking access to the active site. So they kind of get in the way so that the other substrates that want to react with the enzyme can't. And this can be a good thing because you don't necessarily want your enzymes working all the time. So your body has a number of feedback mechanisms whereby if too much of a particular product is formed, it will inhibit the reaction of the enzymes to prevent more of that product from being formed. So, and I'll show you a picture of that in a second. And then there's non-competitive inhibitors. Non-competitive inhibitors. Right? Um, these are molecules, or I'll just say substances that bind to the enzyme in a different location than the active site. If they don't bind to the active site, they bind to the enzyme in a different location. And they cause the active site to change shape and render the enzyme ineffective. So, like for example, and if you let's say you had an enzyme that looked like this, okay? It was designed to fit something that was shaped like that, okay? But it also had a little attachment right there for something else, for the inhibitor to bind. But normally, so the active site would be right here, but a non-competitive inhibitor could distort the active site by binding here. So let's say we had a non-competitive inhibitor. And that came up and attached to it. Okay. And the active site then gets messed up. Yeah. So the active site gets messed up by that non-competitive inhibitor coming in here and binding with the protein and distorting the shape. So initially the substrate might have looked something like this, and it might have been able to fit in the active site, but now you can see that substrate's not going to be able to fit into the active site anymore. It's just not going to work. So that's what a non-competitive inhibitor does. It binds at a different location on the enzyme and distorts the active site and makes it impossible for the active site to be usable or functional. All right. And competitive inhibitors have basically the same shape. They fit the active site perfectly, just like the substrate, but they're not active in the sense that they don't cause a reaction to take place. They just block the site and prevent substrates from getting in there. All right, cool. Any questions over anything that we went over today?
No, I'm good. I kind of did this last week.
Oh, okay. Oh, cool. Well, thank you so much for coming. And y'all have a great week, and we'll see you for the final exam review.
Okay, thank you.